Good morning, students. Students, today I am talking about the last and final microbiomolecules, and these microbiomolecules are basically nucleotides. Now, students, I want to tell you that what are nucleotides and what is their structure, how they are composed of, as well as what are their functions. So, students, initially I want to tell you that these means nucleotides are organic compounds with heterocyclic rings. Means nucleotide is a kind of organic compound which is composed of heterocyclic ring shape structures. ठीक है, a nucleotide consists of nitrogenous base, pentose sugar and a phosphate group. So if you will observe the structure, so you will find out that it is basically composed of three different things. First one is nitrogenous base, ठीक है, second is pentose sugar and third one is phosphate group. Initially I am talking about the nitrogenous bases. So we have two different types of nitrogenous bases in nucleotides. The first ones are called purines and the second ones are called as pyrimidines. ठीक है, if you want to remember what are purines, what are pyrimidines, तो ct pi you can use a formula ct pi cytosine and thymine. ठीक है, these are pyrimidines. ठीक है, while rest of the two adenine and guanine are purines. Now what is the difference between purines and pyrimidines? Now if you will observe, purines are heterocyclic rings, but two heterocyclic rings are fused together in case of the purines. Like you can observe the structure of adenine and you will observe the structure of the guanine. These are the examples of purines, and here you are observing two heterocyclic rings are fused together, while pyrimidines ct pi, as I told you, ct pi cytosine and thymine. But in case of the RNA, instead of thymine there is another kind of nitrogenous base, it is called uracil. So thymine, cytosine and uracil, these three are kept into the category of the pyrimidines, and they have a single heterocyclic ring. ठीक है, these are two types of uh, nitrogenous bases, purines and pyrimidines. What are purines? Adenine and guanine. And what are pyrimidines? Thymine, cytosine and uracil. Is it clear? Now the second is pentose sugar. Now in nucleotides there are two types of pentose sugar which may be present. ठीक है, राइबोज सुगर और डीऑक्सी राइबोज सुगर, दीज आर द टू डिफरेंट टाइप्स ऑफ पेंटोज सुगर्स विच कैन बी प्रेजेंट इन न्यूक्लियोटाइड्स. ठीक है, राइबोज और डीऑक्सी राइबोज. तो इन द ड्यू टू द प्रेजेंस ऑफ राइबोज सुगर RNA is there, due to the presence of deoxyribose sugar DNA is there. Now the third important component of the nucleotide is phosphate. ठीक है, now phosphate group is the third important component. Now except it I want to tell you some other things. ठीक है, actually we can say that what are nucleotides, तो you can say that nucleotides are phosphorylated nucleosides. ठीक है, यानी ऐसे न्यूक्लियोसाइड जिनका फॉस्फोराइलेशन हुआ है, जिनके साथ एक फॉस्फेट ग्रुप जुड़ा हुआ है, उनको हम कह सकते हैं कि ये दीज आर न्यूक्लियोटाइड्स. अब क्वेश्चन मार्क ये आता है कि व्हाट आर न्यूक्लियोसाइड्स. ठीक है, व्हाट आर न्यूक्लियोसाइड्स, तो वी कैन से दैट देयर आर थ्री टाइप्स ऑफ कंपोनेंट इन द न्यूक्लियोटाइड. व्हाट आर दिस? नाइट्रोजीनियस बेसिस, पेंटोज सुगर एंड फॉस्फेट ग्रुप. only if only two of them are attached together or joined together, means what are what two nitrogenous base plus pentose sugar are together, then these are called nucleoside. When phosphate group is going to add with them, then it is becoming nucleotide. So you have to know the difference between nucleoside and nucleotide. When only nitrogenous base and pentose sugar are there in combination, then this is nucleoside. ठीक है, and four we have five different types of nucleosides are there. What are these? If adenine nitrogenous base is added with the pentose sugar, then it is called adenosine. ठीक है, similarly guanine is added with the pentose sugar, then it is called guanosine. If thymine is added with the pentose sugar, thymidine. If cytosine is added with the pentose sugar, cytidine. ठीक है, if uracil is added with the pentose sugar, uridine. If and these are five different types of nucleosides. Now nucleoside plus phosphate group, it is called nucleotide and it is referred as, अगर देखो अब guanosine के साथ ऐड होता है आपका phosphate group, then it converts into adenyllic acid. Similarly guanosine के साथ जब आपका ऐड होता है phosphate group, then it converts into guanylic acid. Similarly जब thymidine के साथ आपका ऐड होता है phosphate group, then it is called as thymidyllic acid. While cytidine के साथ अगर आपका ऐड होता है phosphate group, then it is called cytidyllic acid. वहीं यूरिडीन के साथ अगर ऐड होता है आपका फॉस्फेट ग्रुप, देन इट इज कॉल्ड एज यूरिडिलिक एसिड. तो यू हैव टू रिमेंबर व्हाट आर न्यूक्लियोसाइड्स एंड व्हाट आर न्यूक्लियोटाइड्स. न्यूक्लियोसाइड मीन्स नाइट्रोजीनियस बेस प्लस पेंटोज शुगर. ठीक है, दिस इज कॉल्ड न्यूक्लियोसाइड. और न्यूक्लियोसाइड प्लस फॉस्फेट ग्रुप इट इज कॉल्ड एज nucleotide. Is it clear? So on the basis of two different types of nucleotide, now which kind of sugar is added? ठीक है, either the deoxyribose sugar is added or the ribose sugar is added. ठीक है, there are two types of nucleic acid which can be formed. DNA deoxyribonucleic acid. Now अभी nucleic acid जो बनते हैं, these are formed of polymerization of these nucleotides. ये न्यूक्लियोटाइड आपस में जुड़ करके पॉलीमर बना लेते हैं और ये एक पॉलीन्यूक्लियोटाइड चेन तैयार करते हैं. और ये पॉलीन्यूक्लियोटाइड चेन ही आपकी क्या कहलाती है, न्यूक्लिक एसिड्स कहलाती हैं. और देर आर टू टाइप्स ऑफ न्यूक्लिक एसिड्स विच आर फॉर्म्ड बाय टू डिफरेंट टाइप्स ऑफ न्यूक्लियोटाइड. ठीक है, डीऑक्सी राइबोन्यूक्लिक एसिड एंड राइबोन्यूक्लिक एसिड. दीज आर रिफर्ड एज जेनेटिक मटेरियल इन लिविंग ऑर्गेनिज्म. ठीक है, अंडरस्टूड. नाउ आई एम कमिंग टू द फंक्शंस ऑफ द न्यूक्लियोटाइड्स. ठीक है, न्यूक्लियोटाइड्स आर वेरी इंपॉर्टेंट. ठीक है, प्यूरिन एंड pyrimidine न्यूक्लियोटाइड्स आर मोनोमर्स forming nucleic acids because either the purine or pyrimidine nucleotide, ठीक है, whichever type of nucleotide is there, they will attach together, they will join together and they will form a long polynucleotide chain and finally they will form a nucleic acid. Sometimes what happens with these nucleotides, one additional phosphate groups adds. ठीक है, and whenever the adding of the whenever a new another phosphate group is added with the nucleic acids or nucleotides, this phosphate group attains very much high energy, and whenever the energy is required, this phosphate group or phosphate bonds breaks down and releases a hyper amount or a huge amount of energy. And sometimes some special nucleotides like nicotinamide nucleotide or riboflavin nucleotide act like a coenzyme. Now what are coenzymes we will discuss later on. ठीक है, now here the biomolecule concept is finished. Now I want to tell you that these biomolecules can also be known as metabolites. ठीक है, these biomolecules which we studied earlier on, these can also referred as metabolites. ठीक है, or in another way we can say that all the biomolecules which are extracted from living organisms are called as metabolites. And metabolites can be classified into two different types. ठीक है, and these metabolites are of two different types: primary metabolites and secondary metabolites. Now what is the difference between primary metabolite? Now primary metabolites are those metabolites which are clearly identifiable. ठीक है, विच आर क्लियरली आइडेंटिफिएबल मीन्स जिनको आप देखते ही उनको पहचान सकते हो कि आप उनका क्या फंक्शन है, आप आइडेंटिफाई कर सकते हो. एंड दे हैव ए स्पेसिफिक एंड दे प्ले ए स्पेसिफिक रोल इनसाइड द लिविंग ऑर्गेनिज्म. दीज आर कॉल्ड primary metabolites. So what are primary metabolites? Primary metabolites are those which have identifiable functions and play specific roles in all physiological actions of the cell. ठीक है, फॉर एग्जाम्पल अमीनो एसिड इज ए प्राइमरी मेटाबोलाइट. ठीक है, नाइट्रोजीनियस बेसिस आर द प्राइमरी मेटाबोलाइट. लिपिड्स आर प्राइमरी मेटाबोलाइट्स. ठीक है, नाइट्रोजीनियस बेसिस हैं, प्रोटीन्स हैं, न्यूक्लिक एसिड्स हैं. ठीक है, ऑल दीज आर प्राइमरी मेटाबोलाइट्स. बट एक्सेप्ट देम देयर आर सम मेटाबोलाइट्स टू. ठीक है, इनके अलावा भी कुछ मेटाबोलाइट होते हैं. एंड दीज आर फॉर्म ड्यू टू द वेरियस मेटाबॉलिक रिएक्शंस. तो ये जो सेकेंडरी मेटाबोलाइट्स होते हैं ये एंड प्रोडक्ट होते हैं जो सेल के अंदर चलने वाली डिफरेंट मेटाबॉलिक प्रोसेसेस के तहत बनते हैं. ठीक है, नाउ आई एम गोइंग टू गिव यू सम एग्जांपल्स. नाउ देयर आर वेरियस टाइप्स ऑफ पिगमेंट्स आर प्रेजेंट इन द सेल्स लाइक एंथोसाइन है. ठीक है, कैरोटिनॉइड्स हैं. दीज पिगमेंट्स आर एग्जांपल ऑफ सेकेंडरी मेटाबोलाइट. ठीक है, एंड देयर आर वेरियस ड्रग्स लाइक वीनाब्लास्टिन है और कुरकुमिन है. ठीक है, एल्कालॉइड है, मॉर्फिन है, कोडीन है. दीज आर सम एल्केलॉइड्स. Some essential oil like lemongrass oil, this is the secondary metabolite. Some lectins, these are the secondary metabolites. Some terpenoids, ठीक है, like monoterpenes and diterpenes, these are the examples of secondary metabolites. There are some toxins like aberrin and rixin, these are also the secondary metabolites. And there are various polymeric compounds like rubber है, cellulose है, gum है, resins हैं. ठीक है, and we are using different types of spices. ठीक है, these are also the example of secondary metabolites. So you will be able to identify what is the difference between, because the primary are very easily identifiable and their function is known, as well as they are very specific to perform various physiological functions like amino acid, nitrogenous bases, proteins, nucleic acids. While the secondary metabolites are the products of various metabolic processes like pigments, drugs, alkaloids, essential oils, terpenoids, toxins and polymeric compounds. I hope so you understand the topic. Still, if you have any difficulty, please ask. Till then, thank you, bye bye, and have a nice day.